Okay, so welcome back to this next video in which we are discussing methods used for investigating exocytosis. Okay, so we've just talked about how we can use lipid mixing assays as an in vitro fusion assay for ascertaining as to whether two populations of vesicles have in fact fused. Okay, now the next one that I want to talk about, which is a content mixing assay. The aim is exactly the same. The aim is that we have these two populations of liposomes, and I'm sorry I just said vesicles again, okay? We have these two populations of liposomes, the V snare containing liposomes and the T snare containing liposomes, and we want to see whether they have fused basically, okay? And this is another method to identify whether the two liposomes have fused. Okay, so in this case you don't attach the fluorophore molecule to lipids like we used in the previous case. Instead, the fluorophore molecule is inside the liposome. Okay, and it's going to be inside the V liposomes. Okay, so the V snare containing liposomes. So here is our liposome, our V snare containing liposome, and basically we are going to put inside this liposome a fluorophore known as HPTS. Okay, so this is capable of absorbing a certain frequency of electromagnetic radiation, and then it will emit another frequency of electromagnetic radiation, which will be lower than the excitation frequency. So if I just draw that on here, we will put some excitation frequency in, and we'll get out some emission frequency. Okay, so it has the property of fluorescence. Now, HPTS's fluorescence properties can also be quenched, okay, but in a different way to the way that MBD was quenched by rhodamine. Okay, this quenching is not going to work using fluorescence resonance energy transfer. However, you can put in a molecule into the lumen of these liposomes, known as DPX, okay, which is capable of stopping the HPTS from fluorescing basically, and it does it by a mechanism other than FRET. Okay, so it's not that it's absorbing the emission frequency of radiation here, it does it by another mechanism. Okay, so it interacts with the HPTS and stops the HPTS from emitting this radiation basically. So the idea is that originally in the V snare containing liposomes, what you do is you put in this high concentration of HPTS and you also put in a high concentration of DPX, and then you get no fluorescence basically, or very little fluorescence from the HPTS, okay, when you shine the excitation frequency for HPTS on these V snare populations of liposomes. Okay, then what you can do is you can mix the V snare containing liposomes with the T snare containing liposomes. So I'll draw this here. Here is our container, and we will now mix V snare containing liposomes and T snare containing liposomes, and there will be absolutely loads of these things in this container. Okay, and the idea then is that if V snare containing liposomes and T snare containing liposomes do indeed fuse, then what will happen is the HPTS and DPX will become diluter, okay, when they mix, well, when the two liposomes fuse. Okay, so now HPTS and DPX will be more dilute in this fused liposome that we now have, and that means that each HPTS molecule is on average going to be further away from a DPX molecule, so the chance of the fluorescence getting quenched by the DPX molecules is going to be decreased basically. So now when you shine electromagnetic radiation of the excitation frequency for HPTS onto the medium, what you're going to find is that the fluorescence signal goes up, i.e. the amount of electromagnetic radiation of the emission frequency for HPTS that you're getting back is going to increase, and that's a way then that you can tell that T snare containing liposomes and V snare containing liposomes have fused, and because it's mediated by the content of the liposomes, it's called a content mixing assay because it relies on the content of the two liposomes mixing. Okay, right. Now once again, you want to be able to tell the difference between which fusion is generated by the snare proteins, and what fusion is snare independent. Okay, and again, you do this using the botulinum neurotoxin D to cleave the VAMP2 on the surface of the V snares. That disables all snare-mediated fusion, so the signal rise that you get even with the addition of the botulinum neurotoxin, that then serves as your control. That's your snare independent fusion, basically. So if I show what this might look like, if we have a graph here, okay, and this is the intensity of the emission frequency of electromagnetic radiation that we're getting back, it might start off very, very low, and then when you mix them with the snare proteins intact, you might get a signal that looks like this, okay, and then it will stay up there, okay, whereas when you mix them with the snare proteins cleaved, you might instead get something that looks like this. So what you'd want to do is subtract this off from the rise that you got here, and that would then be the amount of fusion that was driven by the presence of the snare proteins. Okay, right. Now there's one other technicality that I want to mention here, okay, which is that another problem that could occur, that could cause the fluorescence signal to go up, even though there was no fusion, is that HBTS could be leaking from the from the liposomes here into the medium, and then if it was leaking into the medium, then its fluorescent properties would go up, okay, and that would look as though these liposomes, were fusing even though they weren't. Okay, so what you do is you do this experiment in the presence of a very high concentration of DPX in the medium, so that any HBTS that does leak out of the liposomes is just going to get quenched immediately, and therefore it's not going to contribute to any sort of signal that you can detect. Okay, right, so that's another technicality of the content mixing assay for in vitro fusion. Right, okay, so what we're going to turn our attention to now is looking at imaging techniques. We want to look at ways whereby we can actually watch exocytosis occurring within cells. Okay, and this is specifically going to be very useful in those adrenal chromaffin cells, but remember that the adrenal chromaffin cells' mechanism of exocytosis is the same as the exocytotic mechanism of the axon terminals of neurons. Okay, so basically the idea here, and I'll just put a title, we're going to be doing imaging here. The idea here is that we can get the chromaffin cell, so I'll draw my adrenal chromaffin cell here, okay? What we can do is we can get the adrenal chromaffin cell to express a molecule that it will put into its vesicles that will be fluorescent. Okay, and what we usually do is we use neuropeptide Y, which has had green fluorescence protein attached onto it. Okay, so we make a fusion protein, which is mainly neuropeptide Y, but then right on the end of it, it has green fluorescence protein sticking off the side. Okay, now the key thing about green fluorescence protein is that it's a fluorophore. You can send a certain frequency of radiation in to excite green fluorescence protein, the frequency of excitation, and you'll get another frequency of radiation out, the emission frequency. Okay, so green fluorescence protein is a fluorophore. It's a protein that acts as a fluorophore, and we can attach it onto other molecules which will get loaded into synaptic vesicles, or in this case, secretory granules, since we're talking about an adrenal chromaffin cell here. Okay, so this is a secretory granule. Okay, so this molecule that I've attached the green fluorescence protein onto, this is what's known as neuropeptide Y, which is a neurohormone molecule, okay, which will be loaded into the secretory granules. Okay, so basically the idea then is that we can take these adrenal chromaffin cells, we can make them express this neuropeptide Y with green fluorescence protein attached onto it. That will end up stored in the secretory granules, and then the idea is that we have a way of watching these secretory granules. However, there is still a problem here, okay, and the problem is that if I now shine electromagnetic radiation of the excitation frequency for green fluorescence protein onto this cell, Okay, it's going to go to all different depths of this cell. It's going to go all over the place. Okay, and I'm going to be getting emission frequency radiation coming back at me from all over the place. So there's a risk that all I'm going to get is a great big green blob coming back at me. This is what I'm going to see if I look at this, okay, with a microscope. Okay, that's the risk, that's the problem. Okay, now you might ask, well, why are you going to see that? Well, if you think about it, I'm going to see, if I look from one aspect, I'm going to see light coming from all different levels of the cell, all different layers of the cell, and potentially there's secretory granules all over the place at different layers, basically. Okay, so this is green light coming from secretory granules that are at different layers that are now appearing stacked on top of one another. So if I try and draw a three-dimensional spherical picture to explain this, the cell we'll imagine is a three-dimensional sphere like so. You're going to have secretory granules all over the place at different layers in this cell. Okay, so it's likely that if I just look at a two-dimensional picture, if I'm looking at, from one angle at this cell, I'm going to end up just seeing a great big green blob, because at some level, okay, some point through the cell's width, you're going to have a secretory granule. Okay, so that's a problem. What we really need to be able to do is just look at a thin slice of the cell. We need to be able to take a cross-section through and just look at a thin slice where you'll just have a cross-section of a few secretory granules. Okay, now how can you do that? Well, there is a technique by which you can do that, whereby you can excite only the green fluorescence proteins in a little thin slice of the cell, and this is what's known as total internal reflection fluorescence microscopy, TIRFM. So what this stands for is total, that's the T, internal, that's the I, reflection, that's the R, okay, and then fluorescence microscopy, okay, fluorescence microscopy, and this uses the phenomenon of total internal reflection. Okay, right, so I'm going to now describe how this process works on another page. Okay, so basically this is the idea. What you're going to do is you're going to have your glass slide here. Okay, so this is the glass slide, and then you're going to have your adrenal chromaffin cell sitting on top of the glass slide here. And the idea is we're now going to look at a tiny little slip of the cell, okay, around 10 nanometers, sorry, not 10 nanometers, 100 nanometers thick. We're just going to look at this tiny little section of the cell using this technique of TIRF microscopy. Okay, now how is it going to work? Well, basically, it relies on a phenomenon known as total internal reflection. So let's discuss total internal reflection, which is often just abbreviated down to TIR. So basically, total internal reflection is a weird phenomenon that is in the natural world, okay, whereby when you shine light onto an interface like this, okay, where light is moving from one substance which has a high refractive index to an area which has a low refractive index. And let me just explain what refractive index is, because refractive index is a really important concept for understanding this. Okay, so refractive index tells you how much slower light moves through a substance than it would move through a vacuum. Okay, and it's usually abbreviated as N. So basically, if your refractive index is 7, let's say, which is a very, very high refractive index, I don't know if there are any substances where a refractive index is 7, what it would mean is that that substance, through that substance, light moves 7 times more slowly than it would move for a vacuum. Okay, so we all know that the speed of light for a vacuum is the famous constant, 3 times 10 to the 8 meters per second. Okay, which is incredibly fast, and supposedly nothing can go faster than that. Okay, so that's the speed of light through a vacuum, so through empty space, that's how fast light moves. However, when you shine it through something like glass, it moves slightly slower than that. okay? And how much slower it moves through that substance is known as the refractive index of that substance, okay? So a high refractive index means that the light moves much slower than it would through a vacuum. A low refractive index, closer to 1, the ideal refractive index is 1, which means that light moves at the same speed through you as it would move through a vacuum, okay? A lower refractive index, which is towards 1, would mean that the light really moves very close to the speed of light in a vacuum, basically. It's not that much slower. Okay, so basically the idea here is that the glass slide here has a high refractive index, and then the cell cytoplasm has a lower refractive index. Okay, so what we're going to do is now shine laser light on like this through the glass here, and then when the laser light gets to the point of moving from the glass to the cell cytoplasm, it's going from an area of high refractive index to an area of low refractive index. Basically, the phenomenon of total internal reflection is that if this angle at which you shine the light on, this angle here, which is known as the angle of incidence, so theta here, this is the angle of incidence. If this angle of incidence is greater than a certain critical value, the light will not move from the glass into that cell cytoplasm. Instead, it will reflect off. Okay, so if the angle of incidence, which is theta, is greater than the critical value, okay, or the critical angle as it's called, then what will happen is the light will not move through the glass into the cytoplasm of the cell, but instead is going to completely, totally internally reflect and will come off like so. So if you shine photons onto this interface between the glass slide and the cytoplasm of the cell, and you do it beyond this critical angle, then they will totally internally reflect off. And the critical angle will depend on which frequency of light you send. Notice I have not told you which frequency of light this is, okay? That's why I said it was laser light. I said laser light, and what that means is that all of the photons that you are going to be sending, like so, are going to be the same frequency. That's the characteristic of laser light, that all the photons that are being sent will be of the same frequency. So all of them will have the same critical angle, basically. They will all totally internally reflect if the angle of incidence is greater than the same critical angle. But that critical angle does depend on which frequency you're using, which is why it's important to be using laser light here. Otherwise, all the different photons would need a different critical angle to actually display this phenomenon of total internal reflection. Okay, so that's the phenomenon of total internal reflection, which is that for laser light, i.e. light of a certain frequency, if the angle of incidence is beyond a certain critical angle, the light will not move from the area of high refractive index into the area of low refractive index. Instead, it will totally internally reflect off. Now, for reasons that I'm not going to pretend to understand, some of the energy of the light doesn't actually get totally internally reflected. Some of the energy propagates on forward from the surface of the interface here into the cytoplasm of the cell. Okay, so some energy moves on forwards. Okay, some sort of disturbance in the electrical and magnetic fields propagates on forwards. Okay, and this propagation of energy on forwards is what's known as an evanescent wave. Okay, and basically the idea is that the energy of this evanescent wave can then be absorbed by our green fluorescence protein, okay, if it's of course of the right frequency radiation that we use here, and will excite the green fluorescence protein, and then the green fluorescence protein will release its frequency of emission radiation, basically. Now, the important thing then is that this evanescent wave does not go very far. It usually goes around 100 nanometers into the cytoplasm of the cell, and then after that it really doesn't get any further. Okay, now how far the evanescent wave does penetrate is dependent upon the angle at which the light is incident, basically. If you increase the angle of incidence here, then basically the penetration goes down. So if instead I fired the light like so, and it totally internally reflected like that, the penetration of the evanescent wave caused by this would be lower, basically. So penetration depends on this angle of incidence here. Okay, right. So the idea here is then that we can excite only the green fluorescent proteins that are in this very little slip here, and then all we need to do basically is have some camera taking pictures, basically, some microscopic camera here taking pictures of this and looking for the frequency of emission of green fluorescence protein, and now we get around the problem of seeing this light coming from all different levels of the cell. We're only going to see it coming from this base of the cell here, and of course the base of the cell is going to contain some synaptic vesicles, or rather some secretory vesicles, like so, attached and primed to the plasma membrane of the adrenal chromaffin cell. So here they are. They'll have green fluorescence protein in, so they'll appear green. So we are now going to get a beautiful picture of secretory vesicles attached to the plasma membrane through this technique. So the whole purpose of the TIRF microscopy is just so that you can illuminate a very thin slice of your cells, okay, to get around this problem that you are seeing emission frequency radiation coming from all different levels of the cell, and therefore it just appears as a great green blob. Okay, now you can see individual vesicles, basically. Okay, and it works through this phenomenon of total internal reflection by creating these evanescent waves. So you need to match the frequency that you use here such that this evanescent wave is going to be able to excite the green fluorescent proteins that are in the secretory vesicles of the adrenal chromaffin cells. Okay, right, so that's TIRF microscopy, which is a very important imaging technique. In the next video, what we will do is we'll then talk about two final techniques which can be used to measure vesicle exocytosis, which are using capacitance measurements and also using amperometry.